Toxins are poisons from biological organisms. Example, aflatoxin, which is produced by Aspergillus fungus, which is a fungus which live on corn and nuts. So this aflatoxin causes liver cancers. Xenobiotics are synthetic chemicals. For example, pharmaceutical chemicals or TCDD, okay, which is commonly known as dioxin. It is produced during production or combustion of chlorinated organic chemicals. Okay, and this dioxin or TCDD is well known for causing chloracne. Toxicants are the poisons or toxins or the poisons that are not produced by biological organisms. Okay, for example, lead is a toxicant, polyaromatic hydrocarbons is a toxicant, or arsenic is a toxicant. Okay, so the difference is very obvious. Toxins are produced by biological organisms, so toxicants are either man-made or naturally occurring, but they are not produced by biological organisms. Xenobiotics are again are the chemicals such as pharma and dioxins, which are not produced by biological organisms, but these are external to body. So something that is from outside the body, a chemical from outside the body that enters a body is xenobiotic. Let's look at some common toxins or poisons or toxicants that and their mechanism of action. Botulinum toxin, botulinum toxin is acted by presynaptic binding and thereby decreases the ACH release, acetylcholine release, leading to neuromuscular block. Then TCDD, which we have already seen. Sorry about that. TCDD is acted by binding to aromatic hydrocarbon receptors. These are intracellular receptors. Then tetrodotoxin, it is voltage gated sodium channel blocker. By doing so, it what it does is it inhibits the firing of action potential and leading to neurotoxicity. Okay, then there is hemicolinium 3. Okay, hemicolinium 3, what it does is it blocks the choline reuptake. How? By acting on acetylcholine transporters. Okay, these are presynaptic transporters that continuously take the choline that is formed by breakdown of acetylcholine in synapse. Then the next common chemical or well-known toxin is XV nerve gas. Okay, so this is acetylcholinesterase inhibitor, which increases acetylcholine between neuron and muscle. Then strychnine sulfate. This acts by antagonizing, or it is glycine or acetylcholine receptor antagonist. It acts on, it show its effects on via these mechanism. It show its effects on motor nerve fibers in spinal cord. And then chlorpyrifos is another well-known toxin. Whether it's a toxicant, it's not a toxin. It is made by human beings, not biologically produced. Chlorpyrifos binds to acetylcholine esterase and prevents its breakdown. Okay. Then phenobarbital sodium, a well-known chemical. Phenobarbital sodium, it's a barbiturate. It acts by increasing chloride channel opening time. How? By acting on GABA A receptors. Thereby, so as chloride channels are open for more time, naturally it depresses the nervous system. Okay. Phenobarbital, the glyphosate is again and one of the well-known toxicants. It's selectively toxic to plants. It acts by inhibiting EPSP pathway. Synthase, it inhibits EPSC synthase, thereby leading to blockage of shikimic acid pathway. Okay. Okay. So you'd have heard about saccharin. Saccharin is an artificial sweetener. It causes bladder cancers in rats, but not in humans. Okay, because the mechanism of action of saccharin is different, and saccharin causes bladder cancers only at very high concentrations in urinary bladder, which can produce crystals, or the concentration are high enough to cause recrystallization in rats. So these crystals produce constant irritation, thereby causing cancers. But attaining such a high concentration of saccharin in urinary bladder of humans is non-attainable, or it is very difficult to attain even at a very higher intake of saccharin. Okay, so that's why the saccharin is selectively carcinogenic in rats, but not in humans. Thalidomide is a well-known teratogenic chemical. Okay, so uh, it causes teratogenicity by various mechanisms, and it is also found to block the expression of certain genes responsible for angiogenesis. Angiogenesis is nothing but blood vessel formation. So, uh, that's why it is currently being used in treatment of leprosy and multiple myeloma, because pathogenesis of these diseases involves the formation of new blood vessels, that is angiogenesis. But this is not used in pregnant women because of the obvious teratogenicity. So there is going to be a big topic dedicated to thalidomide itself in the other videos. So stay tuned. Let's look at some regulatory setup uh, in USA. So FDA enforces FFDCA. Next, EPA enforces FIFRA. TASCA, RCRA, Safe Drinking Water Act, Clean Air Act, Food Quality Protection Act of 1996. I'm not going to say full forms for each and everything because it is time taking. I'm just going to give a brief notes about these acts. That's all. And then Frank R. Gutenberg's Chemical Safety for 21st Century Act. Chemical Safety for 21st Century Act. This is a recent one introduced in 2016. So, according to this Frank R. Gutenberg Chemical Safety Act, all existing chemicals should be evaluated for their toxicity or safety with strict timelines, and the risk-based safety standards should be updated. To be updated. And also the greater utilization of alternative testing methods. Which leads to decreased use of animal uses in toxicity testing. Okay, and this happens with increased use of computational sciences, or computational toxicology, which is important for tox 21 or tox testing in 21st century initiative taken by ETB, taken initiative taken by NTP, FDA, and NIEHS. Okay, as well as National Center for Advancing Translational Sciences. Okay, so these four: NTP, FDA, NIEHS, and National Academy, National Center for Advancing Translational Sciences. So these four together started this Talks 21 initiative. EPA also enforces CERCLA, it is well known, which is also called as Superfunds Act, or Superfund Authorization or Superfund Amendments and Reauthorization Acts, or SARA. Through this, it gives direction and financial support for what it does. It gives direction and financial support for cleaning up of waste sites. OSHA is responsible for safe and healthful conditions at workplace. Okay, OSHA deals with safe and healthful conditions at workplace. It is implemented by Department of Labor. And there is this one, NIOSH, NIOSH, related to CDC. What it does is it conducts research and make recommendations for prevention of workplace related injuries. Okay, and there is CPSC, which is Consumer Product Safety Commission, and the main work of CPSC is to protect consumers from household chemicals. Department of Department of Transport take care of the hazards related to transport of chemicals, hazardous chemicals. And there is NRC, Nuclear Regulatory or Nuclear Regulation Commission, which regulate civilian use of nuclear materials. Okay, only civilian use, not military use. And SOT, Society of Toxicology, developed ethics or code of ethics for its members. So these are, I just simply touched upon various points that are important. Okay. Now, coming back to The toxicology, sildenafil. It was originally introduced to decrease blood pressure in patients, but the sustained erections was found to be a side effect. So it is being reintroduced, or it was reintroduced to treat erectile dysfunction. Brand name Viagra. For chemical allergy, to occur, or simply chemical allergy by definition itself is a response to a previous exposure. So previous exposure is a precondition. Chemical allergy is also known as hypersensitivity or sensitization. So once a person is sensitized with a particular chemical, even at the lower doses, reactions can be triggered. So haptens are the chemicals, are these sensitizers that enter the body. These haptens combines with internal proteins, forming hapten protein complex. So this complex acts as an antigen because this can be recognized by our immune system. Sometimes the chemicals or haptens are too small to be recognized. So as and when it combines with the internal proteins, the size will increase, and thereby this can be recognized by immune system, and that's when it can be considered as an antigen. So within 1 to 2 weeks, the human body forms antibodies. So the next exposure causes sensitization. The reactions that are so severe that could lead to fatal anaphylaxis. Pattern of allergies varies between animals. In humans, mostly skin and eye allergies can be seen. Okay, skin allergies in form of dermatitis or urticaria or simple itching. Eye allergies such as conjunctivitis. Whereas in guinea pigs, the most common form of allergy is bronchial constriction or asthma. This can also be seen in humans, but most often it is the most common form of allergy in guinea pigs. Let's look at idiosyncratic reactions. These are the abnormal reactions of any individual to a chemical because of genetic or other sensitive factors. Okay, the genetic or other sensitive factors make certain individuals, these occur very rarely in only few individuals, which these factors make individuals susceptible for a very severe reactions. It's not, I think, right to say the severe reactions. The quality of reaction is same, quality of response is same, but these individuals, because of these sensitive factors, respond at very lower doses. And these idiosyncratic reactions could mount immune response via human leukocyte antigens and T cells. Most often, these idiosyncratic reactions are observed in skin, liver, hematopoietic system, sorry, hematopoietic system of immune system. In skin, these idiosyncratic reactions can be seen as maculopapular rashes, okay, or urticaria, or drugs. This is nothing but drug reactions with eosinophilia and systemic symptoms. Drug reactions within eosinophilia and systemic symptoms. And TEN, toxic epidermal necrosis, or Steven-Johnson syndrome. These are some examples of idiosyncratic reactions that occur in skin: maculopapular rashes, urticaria, DRUS, TEN, and Steven-Johnson syndrome. Okay, and some chemicals that are known to cause these reactions are dinitrobenzene, chlorobenzene, piperacillin, bacavir, allopurinol, nevirapine, carbamazepine. Okay, so these are the chemicals that were known to cause idiosyncratic reactions to skin: dinitrochlorobenzene, piperacillin, abacavir, allopurinol, nevirapine, and carbamazepine. Liver-related idiosyncratic reactions are mostly hepatocellular or cholestatic in nature, and some chemicals that are known to cause idiosyncratic reactions in liver are halothane, nitrofurantoin, amoxicillin, and clavilonate, the combination, flucloxacillin. Hematological idiosyncratic reactions includes effects on neutrophils, platelets, and RBCs. Okay, heparin, alpha methyl dopa, and chloramphenicol are known to cause these hematological idiosyncratic reactions. HLA, or human leukocyte antigen, is a genetic variant um, uh, that encodes MHC, major histocompatibility complex. Okay, so there are many types of genetic variants of HLAs. For example, HLA B star 5701. Okay, B star 5701. So occurrence or existence of this variant, genetic variant of human leukocyte antigen, is a risk factor for hypersensitivity reactions, from abacavir. So we have already seen that abacavir is something that causes idiosyncratic reactions in skin. So the presence of this human leukocyte antigen 5701 is reason for the abacavir's idiosyncrasy in skin. Okay, similarly, uh, flucloxacillin, we have already seen that this causes idiosyncratic reactions in liver. So again, this is the reason for this genetic presence of this genetic variant is the cause for causing idiosyncratic reactions in liver by flucloxacillin. And then there is another genetic variant, HLA P star 1502. Okay, 1502 is again a risk factor for Steven-Johnson syndrome. Steven-Johnson's syndrome. So we have seen this as one of the idiosyncratic reactions to skin, but the presence of this genetic variant of human leukocyte antigen P star 1502 causes Steven-Johnson syndrome as a side effect by carbamazepine. Okay, and not only this human leukocyte antigen genetic variants, but also the variants of glutathione S transferase are also involved in causing idiosyncrasy. Okay, sarin is a nerve gas. Similarly, hydrogen cyanide. So these two chemicals are known to show the immediate toxicity. Okay. They interfere with cardiovascular respiratory and neurological functions, thereby causing immediate death. What are Sorin and HCN? So these interfere with all vital functions, cardiovascular, respiratory, and neurological. Whereas DES, diethylstilbestrol, is a type of estrogen or synthetic estrogen. So it was given to the pregnant woman to prevent preterm birth. Okay, to prevent this, they used to prescribe diethylstilbestrol, but as a side effect, they found that in the daughters born to the pregnant woman taken diethylstilbestrol had vaginal cancers in young adulthood during 20 to 30 years of age. Okay, so the in utero, sorry, so the in utero exposure, or the exposure while in the mothers, or they were still embryo, caused the toxic effects around 20 to 30 years later. So this is an example of a type of delayed toxicity. Okay, some organophosphates which are used as insecticides are known to cause what is known as delayed neuropathy. So they cause delayed neuropathy by covalent modifications on NET enzyme. Okay, nothing but neuropathy target esterases. Sorry, it is NTE, not NET. Neuropathy target esterases. So these are esterases. These are enzymes. What these organophosphate insecticides does is they bind to NTE covalently, and they thereby eventually causing degeneration of long axons in peripheral nervous system and central nervous system. One example of such organophosphate that could cause this delayed neuropathy is triorthoglycyl phosphate. Okay, so in this type of delayed neuropathy caused by organophosphate insecticides, the symptoms were not seen for several days after exposure. So this is one example of delayed toxicity. Liver and gastrointestinal tract has high ability for regeneration. Okay, so they could easily reverse the toxicity caused by various chemicals. They could easily reverse the toxicity. Okay, acetaminophen, which is paracetamol, cause hepatotoxicity. We all know that it is toxic to liver. Even paracetamol causes hepatotoxicity. Within days of the liver damage, the liver can proliferate the remaining centrilobular hepatocytes and oval stem cells, leading to regeneration of liver. Okay, so it can proliferate centrilobular hepatocytes and can differentiate oval stem cells. So that is one example of how regeneration and reversing of the toxicity can occur in liver. Similarly, for GIT, GIT is toxic, is resistant to anti-cancer drug or cytotoxicity of anti-cancer drugs in general because of the rapid proliferation of epithelial or cryptepithelial cells. These are the cells that line the inside of intestine. Okay. So these are few examples. For example, liver and GIT are damage caused to liver and GIT can be easily reversed because of regeneration capacity of these organs. Whereas the damage caused to nerve cells, for example, damage caused in CNS is almost irreversible. It's a kind of permanent damage. For example, methylphenylpyridinium causes neurotoxicity. MPP plus causes neurotoxicity. This chemical, it's a metabolite of MPP. This chemical selectively targets dopaminergic, that means dopamine neurons. Okay, where in substantia nigra pars compacta, neurons in substantia nigra pars compacta. So this is one region of brain. We will look into more detailed explanation of various regions of the brain and how various chemicals can selectively damage the regions in the subtopics. But in this introductory chapter, we are going to just see how various toxicities can occur, reversible versus irreversible. So this is taken as an example for irreversible or permanent type of toxicity. Okay, so by selectively damaging dopaminergic neurons in substantia nigra pars compacta, this MPP plus can cause neurodegeneration and clinical signs such as slow motion, which is known as bradykinesia. Okay, so which is known as bradykinesia to cogwheel rigidity. Okay, so that's one story. So this thalidomide, which we have already talked about, caused phocomelia. It's a condition uh, where the kids was born without limbs, only small limbs were there, very small ones. Okay, so this was the condition, phocomelia. Next, local toxic effects occur at the site of contacts. Okay, the site of first contact. For example, chlorine gas, when it enters lungs, it damages the lining and cause swelling. And this could also be fatal. Okay, so this is one example of fatal local effects or local effects in general. Tetraethyl lead. Tetraethyl lead cause local skin effects at the absorption sites. Okay, and also this also have systemic effects. So lead is very toxic. So it causes damage to central nervous system and other organs. Next, severe acid burns eventually may lead to kidney damage. Okay, so this is an example of indirect systemic effect from local effects. Okay, acid burns leading to kidney damage. In toxicology, the target organs are not always the sites of the highest concentration of toxic chemical. Okay, these are not always the site of highest concentrations of toxic chemicals or toxic concentrations of the chemical. Okay, for example, lead accumulates in bone, but lead causes soft tissue toxicity, as we have already seen, brain. Okay, lead acts as a reservoir. Bone serves as a reservoir for lead, and it absorbs and slowly release lead, leading to continuous exposure, thereby continuous toxicity. So as these bones act as reservoirs, once a person is exposed to lead toxicity or neurotoxicity can even be manifested after years of the initial exposure or the exposure. Okay, so that is one organ of that is one example of how target organs are not always the sites of highest exposure, highest concentrations of the toxin. The other one is DDT. So dichlorodiphenyl trichloroethane, it accumulates in adipose tissue, fat tissue, but it is damaging or causes it shows its toxicity mainly in brain and female reproductive system. Okay, so there is this very dangerous example of how the delayed toxicity of DDT or accumulation of DDT could be fatal. For example, the migrating birds accumulate DDT in fat of their body, but when they remigrate or when they start to fly back to their homes, slowly this fat starts to burn to provide energy to the flying birds, and thereby slowly releasing DDT into blood, leading to mortality or death from neurotoxicity, or you know general decrease in reproductive capacity. Additivity or additivity of toxicity of two chemicals uh, is, if for example chemical A causes 2 points of toxicity and chemical B causes 2 points of toxicity. What additivity means is together they cause 4 points of toxicity. Okay, so example is inhibition of acetylcholinesterase by organophosphate insecticides. Then we have the synergistic effects. That means two chemicals that has 2 points of toxicity each could lead to 20 points of toxicity if they together are manifesting toxicity at the same time. So together their toxicity would increase by many folds. Okay, for example carbon tetrachloride and ethanol, they are separately hepatotoxic, but when combined they are very hepatotoxic. Next, potentiation. That means a chemical is not toxic by itself, but when it is combined with other chemical that is toxic, could lead to the increased toxicity of this chemical that is toxic. Okay, so example for that is isopropanol, which by itself is not hepatotoxic, but on combination or combining it with carbon tetrachloride, uh, it it makes carbon tetrachloride much more hepatotoxic. Okay, so that is one form of drug interaction or interaction between various toxicants or toxins. uh, Antagonism. In antagonism can be exemplified by this. Something was okay. For example, a particular chemical causes 4 points of toxicity and other causes 6 points of toxicity. So generally, together they are expected to cause 10 points of toxicity, but whereas when combined together they cause only 8 points of toxicity. So this is one form of antagonism. And then we have this one. Some causes 4 points of toxicity, but the other one causes the opposing effects, thereby nullifying the toxicity of the first chemical. This is one type of other type of antagonism. This can be exemplified by use of the blockers. Okay. For example, naloxone. Naloxone is uh, is used to treat the respiratory depression caused by morphine or morphine-like drugs. Because naloxone is competitive antagonist of morphine receptors, and this can be sometimes life-saving. Similarly, similarly, tamoxifen. Tamoxifen is anti-estrogen, or rather, I would say it's a partial agonist, only partial agonist of estrogen receptors. So uh, it lowers the breast cancer risk in women uh, with high estrogen related carcinogenic risk. Okay, so in the women with high estrogen related carcinogenic risk, this chemical tamoxifen, which is only a partial agonist rather than a full agonist like estrogen, is caused to decrease the breast cancer risk. As a partial agonist, what it does is it binds to estrogen receptors and thereby it blocks the ability of estrogen to bind to estrogen receptors to show its effects. Similarly, atropine is used to treat organophosphate insecticide poisonings because atropine blocks acetylcholine receptors. Okay, then so all these examples that we have seen so far, atropine, tamoxifen, and naloxone. Okay, tamoxifen, naloxone, and atropine are examples of receptor antagonists. Uh, there are also chemical antagonists. Okay, uh, what how this happens is two chemicals interact with each other, leading to less end toxic effect. Okay, for example, DMSA, which is dimercaptosuccinic acid succimer. Okay, dimercaptosuccinic acid, which is also called as succimer. It chelates arsenic, mercury, and lead, thereby decreasing toxicity of these chemicals. And similarly, protamine sulfate, which is a strongly basic low molecular weight protein. Okay, this protamine sulfate forms stable complex with heparin, thereby eliminating the anticoagulant effect. Of heparin. Okay, so these two are example of chemical antagonism: dimercaptosuccinic acid or succimer or protamine sulfate. And there is dispositional antagonism. Okay, in this type of antagonism involves the involvement of ADME effects. Okay, for example, activated charcoal, decreases absorption, or diuretics or osmotic diuretics, or pH alterations of urine. Warfarin, the parent compound is more toxic. Okay, in case of warfarin, the warfarin itself is more toxic rather than its metabolites. So therefore, the phenobarbital, which is microsomal enzyme inducer, is administered, thereby increasing the metabolism of warfarin, thereby ultimately decreasing toxicity. Okay, whereas parathion, which is organophosphate, parent is less toxic, but the metabolite is more toxic. Therefore, something that decreases the or antagonizes the microsomal enzyme activity is administered. Okay, so this is a chemical called pyperonoyl butoxide, which is microsomal enzyme inhibitor. So what this pyperonyl butoxide does is it inhibits the activity of microsomal enzymes, thereby decreasing the metabolism of parathion, thereby decreasing toxicity of its metabolites. Then there is functional antagonism, where chemicals show opposing physiological action. For example, barbiturate intoxication would lead to decreased blood pressure by its actions on GABA A. Okay, which we have already seen that phenobarbital increases the chloride channel opening time, thereby depressing nervous system via GABA A. So barbiturates decreases the blood pressure by acting on GABA A, whereas intravenous administration of epinephrine or norepinephrine, okay, would lead to vasopressor effects by acting on alpha adrenergic receptors, thereby increasing blood pressure. So these both have physiologically opposing effects. So if during barbiturate poisoning or barbiturate intoxication, giving intravenous norepinephrine, this will neutralizes the vascular bone. There are also several types of tolerances. Okay, there are also several types of tolerances. Tolerance is nothing but the repeated exposure decreases the toxicity response. So dispositional tolerance, again, which include effects on ADME. Example is phenobarbital. Phenobarbital produces tolerance to itself by increasing the liver microsomal enzyme expression. Okay. Then there is chemical or cellular tolerance. This is a type of tolerance that is affected by receptors, decreased number of receptors, or decreased number of mediators such as neurotransmitters. Example, during morphine drug abuse, repeated exposure of morphine leads to beta arrestin 2 binding to opioid receptors, thereby causing desensitization or desensitization of opioid receptors, leading to tolerance. Okay, so here the most important part is beta arrestin 2, how it binds to opioid receptors, thereby desensitizing, leading to tolerance of morphine upon repeated exposure. Thank you for watching.